Okay, recording now, and then I'll turn the record off afterwards, just in case you've got more questions that you'd like to ask outside of the recording. All right, so let's have a bit of a chat about the final exams. So normally, of course, we have one final exam, but that has been split into two parts, given that it's online. This is a suggestion that came from La Trobe, that it's not easy sitting in front of a computer completing an exam online for a long period of time. So the recommendation was to first to add 50% to the duration of the exam, and then to split it into two parts and make those parts available within a 24-hour window. All right, so the two parts of the Living Systems final exam are referred to as part 1 and part 2. You may have seen these pop up on Moodle under assessments. If you scroll right down to the bottom of the assessment section, you'll actually see exam part 1 and exam part 2. All right, so they will both be available on Monday, the 15th of June, as you know, because the exam timetable has been released. And I'll show you that once again before you log off tonight. All right, so here is the window. So on that day, Monday, the 15th of June, the two parts of the exam will be available between 12:01 a.m. and 11:59 p.m. So basically a 24-hour window during which time you can commence either part of those, either part 1 or part 2. You can commence them at any time. But once you start either part 1 or part 2, then the clock will be ticking. That is, you will have 70 minutes to complete each part. So essentially what that means is that you could start part 1 if you like, or part 2. If you want to start with part 2, you could start that one at, say, 10 o'clock in the morning if you want. Complete that one, and then you can wait until 7 p.m. in the evening to start the next part, as long as they're completed within this time period here. All right, so you can start with part 1 or part 2. It doesn't really matter. You don't have to do them in order. Also remember that before the time is up, remember to submit your exam. That is, press that submit button, okay? Not that it will disappear, not that your answers will disappear, but it will show as being not submitted. And that causes all sorts of headaches for me, especially. So it would be really good if you remember to submit before the 70 minutes is up. All right, so the weighting for both parts combined is 30%. And as I said, you can find both part 1 and part 2 of the exam on Moodle. So they just became visible this morning sometime, so you can have a look at them. All right, don't forget that you'll need to have your calculator with you and a copy of the periodic table of the elements. So that's also important. All right, any questions on that so far? Or is that all very clear? All good? What about everyone else? Any questions on that? All good, all clear. Okay, all right, I will move on. All right, let's look now at the exam structure. So this is part 1, which will be all multiple choice questions. Each question is worth 1 mark. There are 50 multiple choice questions. And as I said, you'll have 70 minutes to complete. You'll only be facing 1 question at a time. So that means that you're clicking from one slide to the next, or 1 question to the next, quite a few times during this 70 minutes. And that's one reason why the extra time is given for online exams, because that's not something that you would normally do. in a sit-down exam. And also, of course, you don't have the benefit of reading time. Basically, once you start, you go straight into the questions. So that's also factored into that extra time. All right, so that's part one. If you like to start with multiple choice questions, then start with part one of the exam. All right, part two of the exam consists of both true or false questions and short answer calculations. So true or false, each worth 1 mark. There are 44 questions in total. So that's that section. The next section within part two is 9 short answer questions or calculations. Each of these questions is worth either 2 or 3 marks. Actually, I can be a bit more specific. The calculations are worth 2 marks each. The short answer questions are worth 3 marks each. So just thinking about the calculations, given that they're worth 2 marks each, here's an opportunity for you, if you've got time, to actually write out your working out. So don't just put the answer, put the working out in there. And what this means is that even if you get the wrong answer, but your working out was mostly correct, you could pick up 1 mark out of 2 marks, or even 1.5 marks out of 2 marks. But if you just have the answer written and that answer is wrong, then you get 0 out of 2, if that makes sense. Okay, so use that to your advantage. If you've got time, show your working out. Because what happens often is, the student, will just press the wrong button on the calculator, even though they've got the right formula, the right working out, something wrong in the calculator, and you end up with the wrong answer. So this is important if you want to try and pick up, extra marks, sort of marks by default, even though you've got the incorrect answer. Okay. All right, so that's an important tip for you. And of course, part two is also worth 70 minutes. How can we write working out? Okay, all right, very, very simply, the same way that you've seen some of the working out for calculations in some of your tutorial questions. So if you go back to, which tutorial questions? If you go back to, I think it was chemical reactions, the lecture on chemical reactions. Sorry, the tutorial questions on that particular lecture. Remember that I actually gave you some examples of how to work through a couple of formulas. I gave you some written examples of the actual working out. Yes, that's how you would do it. Okay, so you would actually write out, this is the formula, and this is my working out, and this is my answer. Okay, if you've got any problems with that, by all means let me know, but it's as simple as that. That's how you would write it online, exactly the same way. So you'll have a box that appears online, the same way that you did with the, yes, typing. That's exactly right. You would be typing out your working out. Yeah, absolutely. All right, so that's part two. Any questions on that? On part one or part two exam structure? No, not yet. All right, we can come back to that. All right, so what material would capability? Oh, you don't have to worry about subscripts. There is capability to type subscripts. You don't need to waste time doing that. Okay, yep. I make that assumption. So you don't need to worry about the subscripts or the superscripts for that matter. Okay. All right, lecture material. So you need to revise your lecture material, starting with lecture 7, the chemistry of life, and then proceeding through the remaining lectures for the semester. Okay, so they're the lectures, and I'll just touch on them after this PowerPoint. Okay, so from lecture 7 to 18, that's what you should be revising. Of course, as we said earlier, make sure you revise all your tutorial questions based on lecture 7 onwards, the assessed tutes and the non-assessed tutes. All right, give them equal importance. But also remember, don't try just to memorize answers for each question. Try to understand the concepts, because you'll see that sometimes you'll get questions that are similar, but not exactly the same. Okay, so if you're not understanding the actual concepts that are being covered by a particular question, then chances are you won't be able to answer it correctly. All right, calculations. There's not many calculations in that part 2, but there will be a few. Okay, so I've just summarised them here for you, but I'll also show you these within the lectures when we finish this PowerPoint. All right, some additional tips for you. Study focus. Make sure that you really understand this concept of diffusion. Understand the differences and similarities between just simple diffusion across a biological membrane versus facilitated diffusion or facilitated transport. So really make sure you've got a really solid grasp on these concepts. Also, membrane transport proteins, that is channels versus carriers. What are the differences, similarities and differences? And make sure you've got a very good understanding of the concept of osmosis. All right, so while you're thinking of questions, I'll just share the lectures. Okay. All right, so you should start revising lectures from lecture 7. Then we've got lecture 8, gases, liquids and solids. So within this lecture, and I'll just run through where the formulas are that you should revise. Okay, so definitely this one, the ideal gas law. So make sure you revise PV equals nRT and how to apply that. Make sure you have these 2 universal gas constants handy. Okay. So as someone was saying earlier, it is a really good idea not just to have all your lecture notes with you. In fact, you probably shouldn't have them with you. You can have them nearby, but try not to depend on them. It'll take you too long to be looking up, you know, various concepts or formulas and so on in your lecture notes. It's a far better idea to have just a few pages with summaries and important points on them. And you should be able to come up with those summary pages when you've revised all the lectures. Okay, really condense all the lectures into just a few summary pages that you can refer to very quickly, very quickly during the exams. That's would be the preferred method. Otherwise, you will run out of time if you have to keep looking everything up as you go through. All right, so make sure you've got this formula handy, revise how to use it, have these 2 universal gas constants handy as well. Okay, so that's it there. Yeah. Okay, oh, I should say, all right, make sure that you can convert from degrees Celsius into Kelvin. Obviously that goes with this equation as well. All right, next lecture, acids and bases. So we've got a couple of formulas and a couple of concepts. Well, many concepts, but I'm just pointing out the formulas mostly from the lectures. All right, the ion product constant. Here we go. So this is a good one to have handy. Ion product constant. Okay. And the reason that you need to have that handy is that so you can calculate using these formulas. Okay, make sure that you know how to calculate pH given a particular value for the hydronium ion concentration. Okay, and of course, given a value for pH, make sure that you can calculate the hydronium ion concentration. Okay, so the reverse from that. Okay, next, solutions, lecture 10. And we've got a couple of very simple calculations here that most students don't have problems with. And here they are. So here's one. Okay, weight per volume percent concentration. Make sure you can calculate that. And then, of course, this one here, volume per volume percent concentration, which is pretty much the same. Just the units are a little bit different. All right, molarity. Make sure that you can calculate molarity. So in other words, remember N equals C times V. Remember our molarity formula. And sometimes in order to calculate that, you need our molar mass formula. Okay, so make sure you revise these two and have them handy. Unless you've already committed them to memory, that's great. All right, what else do we have here? And of course, we can't forget C1V1 equals C2V2. So that's another good one to practice and have that on hand as well. All right, let's go on to functioning cells. No calculations there. You'll be happy to hear. Movement across membranes. All right, and make sure you're very familiar with the concept of diffusion. All right, here we started talking about a facilitated transport or facilitated diffusion. So make sure that you're familiar with this. Channels and carriers, good one to revise. Osmosis. Be very familiar with this concept of osmosis. So we've got a few slides here. 1, 2, 3, 4, 6. All right, so a few slides here where we're really talking about osmosis. So make sure that you revise all these concepts. And remember, we talked about what would happen to animal cells or plant cells when placed in different solutions, either isoosmotic or hypoosmotic or hyperosmotic. So that's a good one also to revise. Okay, someone seems to be having problems. Okay, they're dropping out. All right, cells and tissues, structure of plants. I'll just move that over. Okay. Genes, chromosomes, and DNA. Lecture 16, genetic code. And I think that is the end of our lecture series and the calculations. All right, so I'm going to open it up for questions, and I'll just stop the record here. So that you can feel free to ask away. Okay, stop recording.